Hello, we are the storytellers of herbal and nutritional mechanisms through scientific research.
Mm.
Welcome to another deep dive where we take a complex topic from the latest research and really try to break it down just for you.
And today, we're diving deep into cannabidiol, CBD. We're looking at its fascinating role as an immune modulator based on a pretty recent scientific review.
That's right. The specific source material guiding our exploration today is a comprehensive article. It's titled Cannabidiol as an Immune Modulator, a comprehensive review.
Mm-hmm.
It was published in the Saudi Pharmaceutical Journal just last year in 2025. And the authors are Khizra Mujahid, Mohammed Shahzaib Rashid, Aska Sabir, Juteik Nam, Tala Ramzan, Wasim Ashraf, and Imran. Quite a team.
Indeed. And our mission, really, for this deep dive is to unpack what this extensive review tells us about how CBD interacts with, well, the incredibly intricate machinery of our immune system.
Right.
We want to explore the sometimes surprising ways this non-psychoactive compound from cannabis sativa might influence immune responses. And we're sticking strictly to the science presented right here in this paper.
Okay, let's get into it. Right off the bat, this review makes one thing really clear. CBD isn't simple. It's not like it just flips one switch, It seems incredibly complex, interacting with a whole network of targets inside our cells.
Absolutely. It's quite different from THC in that regard. THC, has that strong lock and key fit with the classic cannabinoid receptors. CBD's story is, well, it's different. It has a pretty low affinity for those main CB1 and CB2 receptors.
Huh.
But it still manages to influence them indirectly. And importantly, it interacts with tons of other pathways and receptors that aren't even part of that traditional endocannabinoid system.
Wow. So it's like it has a much wider social circle inside the cell. The review mentions things like G-protein coupled receptors. Yes, CB1 and CB2 are in there, but also others like 5-HT1A and GPR55.
Exactly. And ion channels, enzymes. It's quite the network it taps into.
So how do all these different interactions actually add up? What's the potential effect on inflammation and immunity?
That's the core question, right? Let's start with those classic cannabinoid receptors, CB1R and CB2R. CB1 is mostly famous for being in the nervous system, the brain, while CB2 is really prominent on immune cells.
Okay, so if CBD doesn't stick tightly to them, especially CB1, how does the review say it influences them?
That's a great question. So the review notes that even with that low binding, CBD can actually sort of enhance CB1 receptor activity.
Enhance it. How?
Well, not by sticking strongly, but by influencing a key signaling pathway inside the cell. It's called PI3-KAKT. This pathway is super important for things like how cells specialize, differentiate, and also how they manage inflammation. So CBD seems to kind of nudge the system, maybe indirectly through CB1, towards reducing inflammation.
Interesting. And I think I remember reading something about how it also helps our body's own cannabinoids, like anandamide.
Exactly. That's a really crucial mechanism highlighted in the review. Our bodies make these endocannabinoids like anandamide or AEA, and they have natural anti-inflammatory effects. But there's an enzyme called FAAH, fatty acid amide hydrolase, whose job is basically to break down AEA.
Cleanup crew.
Pretty much, yeah. The review explains that CBD inhibits FAAH. So by slowing down that cleanup crew, CBD lets more anandamides stick around for longer. This amplifies its natural anti-inflammatory and neuroprotective signals. It's like giving your body's own soothing system a bit more time to work.
That's clever. So it interacts directly, but also boosts the body's own signals.
Ah.
Okay, now what about receptors outside the usual cannabinoid family? The review mentions the serotonin receptor 5-HT1A. I usually think of serotonin and mood.
You're absolutely right. 5-HT1A is well known for its role in mood regulation, often linked to things like anxiety and depression. The review states that CBD acts as a partial agonist here.
Meaning it activates it, but maybe not fully.
Exactly. Not as strongly as serotonin itself, perhaps. According to the source, this interaction can increase the activity of certain neurotransmitter systems, like serotonin and glutamate systems. And they suggest this contributes to some of the anxiolytic or anti-anxiety effects people observe with CBD. It involves activating these complex signaling cascades using GTP binding proteins.
Okay. What about TRPV1, transient receptor, potential vanilloid 1? That sounds like something to do with heat or pain.
It is. Spot on. TRPV1 is often called the vanilloid receptor or even the capsaicin receptor because it's activated by heat and, the spicy stuff in chili peppers. It definitely plays a role in pain sensation and inflammation. The review points out that CBD is an agonist for TRPV1. It activates it. And this activation influences several downstream pathways, things like COX2, INOS, TNFSHO, NFG, the result being potentially reduced oxidative stress and less production of inflammatory signals called cytokines.
Okay, another non-cannabinoid one and the review mentions is GPR55. What's its deal with immunity?
GPR55 is, well, it's sometimes called an orphan receptor because we're not totally sure what its main natural activator is, though some endocannabinoids might interact with it. The review highlights that CBD acts as an antagonist here.
So it blocks it.
Right. It blocks GPR55 activity. GPR55 is often found near cannabinoid receptors, especially in the brain, but it seems involved in immune signaling too. Blocking it, as CBD does, is noted in this source as a way to help prevent the production of pro-inflammatory cytokines.
And it seems there's also a direct hit on a major inflammation pathway, the TLR4 pathway.
Yes, this is a really significant one discussed in the review. TLR4, toll-like receptor 4, is like a key sensor on our immune cells. It detects signals, say, from bacteria or damaged tissues, and usually triggers a strong inflammatory response.
The alarm bell.
Kind of, yeah. And that response typically involves activating a master switch called NFB. NFBB then tells the cell, okay, produce lots of inflammatory stuff. The review explains that CBD inhibits this TLR4 pathway. It does this by preventing another molecule called A from being broken down. A normally acts like a lock, keeping NFB inactive. By saving AB, CBD basically keeps NFB locked up, which blocks the production of those pro-inflammatory cytokines. It helps keep the lid on that TLR4-driven inflammation.
Wow, that's a pretty clear way it can interrupt things. The review also briefly mentioned CBD interacting with mu-opioid and delta-opioid receptors.
It does, yeah. It notes it acts as an allosteric modulator. That means it doesn't maybe activate or block directly, but it influences how other molecules interact with those opioid receptors. It shows some selectivity, too. The source suggests this could be relevant for things like drug addiction, but it's maybe less central to the immune modulation focus here.
Gotcha. What about those ion channels you mentioned way back? Calcium and sodium.
Right. The review cites studies showing CBD can inhibit certain L-type calcium channels, especially in heart cells. This interaction is linked to potential cardioprotective effects, suggesting CBD might influence heart function, possibly through pathways involving calcium signaling. It also mentions CBD acting as a non-selective sodium channel inhibitor. Basically, it helps stabilize these channels when they're inactive, preventing them from activating inappropriately, which could affect nerve or muscle excitability.
And finally, before we move to cells, what about nuclear receptors, like PPO?
Yes, PPRO, peroxisome proliferator activated receptor gamma. That's a nuclear receptor, meaning it sits inside the cell nucleus and helps regulate which genes get turned on or off. The review specifically links CBD's activation of PPRO to its anti-inflammatory properties.
So?
Activating PPRO is described as decreasing the expression of things like INOS, another enzyme involved in inflammation, and reducing pro-inflammatory cytokine production. This mechanism is highlighted as helping to mitigate inflammation, particularly in cells related to blood vessels and the heart.
Okay, so if we just pause for a second here. What this review is really painting is a picture of CBD as this multitasker, right? It's not just hitting one or two things. It's interacting with this whole complex web cannabinoid system, serotonin, pain receptors, immune sensors like TLR4, ion channels, even nuclear receptors controlling genes.
Yeah.
And it's doing it in different ways, inhibiting enzymes, activating some receptors, blocking others, subtly modulating still others.
Exactly. And it's this multi-target, multi-mechanism profile that the review suggests is the basis for its potential effects. Anti-inflammatory, antioxidant, neuroprotective, anti-anxiety, maybe even cardioprotective. It's definitely not a simple single pathway story. It's a story of complex molecular conversations happening all over the place.
Okay, that helps visualize how CBD is talking to the molecules. Now let's zoom out a bit. How does all that translate to the actual cells of our immune system? The review digs into several different types.
It does. It tries to connect those molecular dots to the cellular level. Starting with lymphocytes, particularly T cells, which are key players in adaptive immunity. The review summarizes studies showing CBD can generally dampen T cell responses.
Dampen them, like make them less active or reduce their signals?
Pretty much. Findings included things like reducing T cell-mediated effects in certain assays, inhibiting their production of specific cytokines like IFN-agatum and IL-17A. These are often involved in driving immune responses. Some of these effects were linked back to CBD influencing signaling pathways inside the T cells, possibly involving that CB2 receptor we mentioned, and maybe how the cells handle oxidative stress.
Okay. Now let's talk about macrophages. These guys are huge players. Cleanup crew, yes, but also major initiators of inflammation. What did the review find about CBD's effect on them? This seems like a big one.
It is, and this section in the review is particularly interesting because it really highlights the complexity we've been talking about and even some, well, conflicting findings. So some studies cited show CBD having a suppressive effect, reducing macrophage production of major pro-inflammatory cytokines like IL-6, IL-1, and TNFi. That sounds pretty clearly anti-inflammatory, right?
Yeah, seems straightforward. But it's conflicting.
Yes, and this is a really important point the review makes sure to emphasize. Other studies looking at macrophages found quite different results. Some showed CBD actually stimulating the production of IL-12, which is usually considered pro-inflammatory, while at the same time inhibiting the anti-inflammatory cytokine IL-10, kind of a mixed signal.
Huh.
And then in different lab settings or using different types of macrophage cell lines, some research even showed CBD increasing the production of a whole suite of pro-inflammatory cytokines, IL-6, IL-8, IL-16, IL-32.
Wow. So the same compound CBD can sometimes seem to reduce inflammation for macrophages and sometimes maybe even increase it, depending on the context, the study, the cells.
Precisely. That's a critical takeaway from this review. The effects of CBD on macrophages seem highly context-dependent. It's not a one-size-fits-all answer. The review also mentions other influences, like modulating how macrophages move, promoting certain regulatory cells called MDSCs, influencing cell death pathways like pyroptosis, and cellular recycling processes like autophagy. But the main headline for macrophages is, it's complicated, and it varies.
That really drives home the complexity we saw at the molecular level. Okay, how about natural killer cells, or NK cells?
NK cells, yeah, part of our innate immune system. They're important for taking out infected or cancerous cells, and they do express cannabinoid receptors. The review summarized findings showing, again, kind of varied effects of CBD on their cytokine production.
Not straightforward either, then.
Seems not. For example, in studies involving psoriasis patients, CBD treatment was linked to reduced IL-17, but an increase in IF-0 from NK cells. Different directions there. Other lab studies showed CBD inhibiting certain cytokines released from activated NK cells, but interestingly, it didn't seem to affect the production of another one, IFN. So again, a picture of nuanced, not uniform effects.
Okay. What about mast cells, the allergy cells, histamine release?
The review does mention some research indicating CBD might influence a marker of mast cell activation and points to maybe the CB2 receptor being involved. However, the review is quite explicit here, noting that dedicated, specific research looking directly at CBD's overall effects on mast cell function is still pretty limited, not much to go on there yet.
Fair enough. And dendritic cells, DCs, they're the ones that present information to T cells, kind of kicking off the adaptive immune response.
Right, crucial messengers. The source notes that cannabinoids in general can trigger programmed cell death, apoptosis in DCs. More specifically for CBD, the review highlighted findings where CBD seemed to promote what's called a tolerogenic phenotype in human dendritic cells grown in the lab.
Tolerogenic, meaning less likely to provoke a strong immune response.
Exactly. It pushed them towards a state where they were less likely to activate T cells effectively. It also changed how these DCs responded to inflammatory signals and the types of cytokines they produced. But the review does add a familiar caveat. The precise mechanisms behind these effects in DCs still need more thorough investigation.
Okay, seems like a theme. Let's move to polymorphonuclear neutrophils, often the first responders on the scene of an infection or injury.
Yeah, the infantry. The review details findings showing CBD can reduce neutrophil accumulation, fewer of them rushing to the site of injury. It can also decrease the oxidative stress they produce. One study linked this, in corneal injury, back to that 5-HT1A receptor we talked about earlier.
Interesting connection.
And it can reduce the formation of NET's neutrophil extracellular traps. These are web-like structures neutrophils release to trap pathogens, but they can also cause tissue damage if they get out of hand. Other studies cited showed CBD reducing neutrophil migration and their pro-inflammatory cytokine production in lung inflammation, possibly through another receptor called the adenosine A2A receptor.
Well, mostly inhibitory effects on neutrophils.
Mostly, but again, context matters. The review also points out a contrasting finding in a different lung model where CBD didn't seem to affect neutrophils, whereas THC did. So still suggesting nuances based on the model or maybe differences between cannabinoids.
Okay. What about the less commonly discussed ones, like eosinophils and basophils, often involved in allergies and parasite defense?
Good question. The review does acknowledge that, yes, the overall endocannabinoid system does seem to influence these cell types, and it mentions that a CBD extract, not necessarily pure CBD, showed some effects in a mouse model of asthma, which heavily involves eosinophils.
But I sense a but coming.
You sense correctly. The review is very clear and explicitly states that within the scope of this particular review article, there is no direct evidence presented to demonstrate that pure CBD itself directly regulates or affects the function of eosinophils or basophils specifically. It really highlights this as an area where more targeted research is definitely needed. We can't assume effects based on extracts or the general system.
Got it. Important distinction. Finally, let's touch on glial cells in the nervous system.
Yeah.
Microglia, astrocytes, oligodendrocytes, not traditional immune cells, but they have huge immune-like roles in the brain, right?
Yeah.
Absolutely critical roles. And the review discusses CBD's influence here too. For microglia, which are basically the brain's resident immune cells, the review notes CBD can influence them potentially through targets like TRPV1, GPR55, maybe CB12 receptors, although, again, that receptor link wasn't always consistently found in the cited studies. Interestingly, CBD was shown to induce apoptosis, that programs cell death, in microglia. In one study, this happened through pathways that seemed independent of the classic CB12 or TRPV1 receptors. Potential links to 5-HT1A, TRPV2, and PPRO were suggested instead.
More complexity.
And CBD was also found to inhibit their movement, their migration, in response to certain stimuli.
We help protect any other glial cells?
Yes, the review mentions findings where CBD protected oligodendrocyte progenitor cells, or OPCs. These are vital for making and maintaining myelin, the insulation around nerve fibers. CBD protected these OPCs from damage and cell death caused by oxidative stress.
And was that through the usual receptors?
Notably, no. As the review points out, this protective effect seemed to happen independently of the commonly studied cannabinoid, phenyloid, TRPV1, or PPRO receptors.
So even within the nervous system, CBD is interacting with different glial cells. Sometimes protectively, but again, the how isn't always clear.
Exactly. The review concludes for glial cells, too, that more research is needed to clarify the precise mechanisms and which receptors are really key in these interactions.
Okay, stepping back from the specific cells, it really feels like the story mirrors the molecular one, doesn't it?
How so?
Well, CBD seems to have wide-ranging effects across many different immune cell types. But those effects are complex. Sometimes they even look contradictory between studies, and they often seem to depend heavily on the specific cell, its state, maybe the surrounding environment, or the experimental setup. The review really emphasizes it's far from a simple CBD is always anti-inflammatory picture at the cellular level.
That's a very accurate summary of the balanced perspective this paper presents. And that inherent complexity naturally leads into talking about, okay, what are the potential clinical implications, but also what are the major challenges and where do we go from here?
Right. The review does touch on potential clinical relevance. It mentions studies exploring CBD in various autoimmune conditions, things like MS, IBD, rheumatoid arthritis, type 1 diabetes, scleroderma, psoriasis, based on all those mechanisms we've discussed, modulating inflammation, affecting immune cells in various ways. It suggests CBD might have potential to help modulate immune responses and maybe alleviate symptoms in these conditions.
Might being the operative word.
Exactly. And it's crucial for you listening to remember this review focuses heavily on the mechanisms, the how it might work part. It doesn't dive deep into detailed clinical trial results for each disease, so we're sticking to that mechanistic level today.
And on a practical note, the review brings up the role of technology in maybe helping CBD fulfill some of that potential. It notes how things like nanotechnology could help tackle some of CBD's built-in challenges, like its poor solubility, it doesn't dissolve well in water, and its bioavailability, meaning how much actually gets into your system and works.
So, tiny delivery systems.
Potentially, yeah. Nanotechnology could offer ways to improve delivery, maybe target it better, and perhaps increase its effectiveness.
But the review is also really direct and quite blunt about the significant challenges and limitations that still exist. Things we need to figure out before CBD could be widely recommended therapeutically.
Absolutely. A major challenge it highlights is exactly what we've been discussing, those inconsistent effects reported on various immune cells. Seeing both potentially anti-inflammatory and sometimes pro-inflammatory responses depending on the context. That makes predicting its effect tricky.
And despite all the buzz and the promising lab science, the authors here are pretty cautious about jumping to clinical conclusions, aren't they?
They are. They explicitly state that, based on the current understanding laid out in their review, it is premature to recommend CBD as a therapeutic agent.
Why premature?
Because there's still too much we don't know about its long-term safety profile, its overall effectiveness in actual human diseases, and critically, how it might interact with other medications people might be taking. Drug interactions are a big question mark.
The review even lists some specific potential adverse effects that have been noted in studies.
It does. Things like potential issues with liver function, hepatotoxicity, potential effects on the nervous system, neurotoxicity, and common things like gastrointestinal disturbances. These need to be taken seriously.
Right.
And those pharmacokinetic challenges we mentioned earlier, they're still big hurdles. Poor absorption when taken orally, it's highly lipophilic, so it tends to get stored in body fat. It undergoes extensive first-pass metabolism where the liver chews up a lot of it before it even reaches the bloodstream.
So less actually gets to where it needs to go.
Potentially, yes. And there's high variability between individuals and how they process it. All makes dosing and predicting effects difficult.
So even at the fundamental science level, there are still gaps in really understanding exactly how it's working.
Definitely. The review points out that while we've identified many potential targets, the precise immunomodulatory mechanisms like the exact roles of TRPV1, GPR55, PPRR, 5-HT1AR, and that enzyme FAAH in regulating inflammation and immune balance still need much more clarity. We see they're involved, but the full story isn't mapped out.
And it sounds like the research hasn't covered all the immune bases yet either.
Correct. The authors specifically highlight a need for more dedicated research on immune cell types that haven't been studied as much regarding CBD effects. They mention B cells, dendritic cells, beyond the initial findings, and certain specialized subsets of T cells to get a truly complete picture of its immune impact.
So what's the path forward? According to the review, what needs to happen next?
Well, it strongly recommends more rigorous, well-controlled studies, especially in humans. That's key. These studies need to carefully compare different ways of giving CBD different administration routes. They need to explore dose escalation, how effects change with different doses, and really pin down the pharmacokinetics in people.
And crucially,
they need to understand how CBD interacts with the specific immune environment of different diseases or infections. Its effect might be very different in someone with chronic inflammation versus an acute infection. They really emphasize the importance of studying these effects in humans to see if all these fascinating lab findings actually translate into meaningful clinical outcomes.
it mentions some planned clinical trials are looking into this, like one in HIV plus patients.
Yes, exactly. Those kinds of studies looking at dose escalation and specific immune markers in human conditions are essential to bridge the gap from the lab bench to the bedside. The review also suggests it's important to study therapies that might combine CBD with THC, as they often occur together naturally, and to conduct long-term safety studies, especially regarding any potential impact on the body's ability to fight off infections if CBD is used chronically as an immunomodulator.
It's such a powerful reminder, isn't it? That while the potential suggested by all these complex mechanisms is genuinely exciting, there's just a huge amount of careful, detailed, rigorous research still needed to translate that potential into treatments we know are safe and effective. This review does a great job laying out that whole complex picture.
It certainly provides a very detailed map of the current landscape, pointing out both the really intriguing possibilities and, just as importantly, the significant unknowns that remain.
Okay. Now, as we start to wrap up, it's really important that we share the specific disclaimer that was provided in the source material itself. So please, everyone listening, understand that.
The information in this research is intended for educational purposes and to support the development of herbal and nutritional compounds into therapeutic agents. It is not a treatment guideline and should not be used as a substitute for medical advice or care.
Definitely keep that in mind. Thinking about everything we've just unpacked from this one really dense review, the sheer number of molecular targets, the varied and sometimes conflicting effects on all those different immune cells, the clear list of challenges still ahead. It just gives you a sense of how incredibly complex the interaction between CBD and our immune system truly is. It's like this dynamic, ongoing conversation happening inside the body, and this paper has given us a significant window into that dialogue. We've covered a lot of ground, I know, but hopefully you now have a clearer picture of some of the key mechanisms and really the state of the science, according to this 2025 review. What stood out most to you? Was there a particular interaction maybe on the macrophages or the effect on TRPV1 that surprised you? We genuinely love to hear your questions about the fascinating and sometimes puzzling science we've discussed today. Feel free to reach out. Stay tuned for the next deep dive, where we continue exploring fascinating scientific research about herbs and nutrients. With us, the storytellers of herbal and nutritional mechanisms through research.